All right, I guess we can go ahead and get started. Welcome, everybody who's online with us tonight in this snowy evening. We've got a nice hearty group of about 7 people here with us at the Nature Center, but more people online. I'm Mark Nolan, Wichita Audubon Society Vice President and Program Coordinator. And just a couple announcements before we get to our talks tonight. So our field trip on Saturday to El Dorado State Park has been canceled due to the cold and the fact the lake may be frozen. There may still be owls out there, but we can go find those another time. Any other announcements, anybody? All right. And so tonight, kind of by happenstance, we have 3 speakers. If you were aware of our schedule originally, we had been planning to have Scott Loss come and talk about his research, and he was unable to come, so he volunteered some of his graduate students at Oklahoma State to come talk with us. So I'll do a brief intro for each of the students, and whatever order y'all prefer to go in after that, I'll let you take it away. So tonight we have Riley Lawson, Holly Todaro, and Dylan Cooper with us. Riley Lawson will speak about a study on Oklahoma State campus testing the effectiveness of feather-friendly window markers in preventing bird collisions. Riley is originally from the Tulsa area and got his Bachelor's of Science in Natural Resource and Ecology Management from OSU in 2020 and returned in 2023 to earn his master's degree. He also, interestingly, interned at the KDWP Education Museum in Pratt, Kansas in 2021. Holly Todaro will be sharing some of her results from her work on the effects of climate change on avian reproduction. Holly is a PhD student at OSU, where her research focuses on how birds respond to anthropogenic threats. She received a Bachelor of Science in Natural Resource Management from Central Michigan University and a Master of Science in Wildlife Fisheries and Aquaculture from Mississippi State University. Holly is originally from Port Huron, Michigan. And we have Dylan Cooper, who will be talking about his work on loggerhead shrikes. Dylan is a current PhD student at Oklahoma State, where he is leading a state-funded research by the Oklahoma Department of Wildlife Conservation with the goals of developing a monitoring protocol for loggerhead shrikes. He has an interesting citizen science component to his work. Dylan grew up in Phoenix, Arizona. He received a Bachelor of Science in Biological Sciences from Arizona State University and earned his master's degree in biology from the University of Nebraska, Kearney. All right, so welcome you guys, and whoever would like to get going first, take it away.
Okay, I can go ahead and go first. So let me just share my screen real quick. All right. Feel free to interrupt me if any like technical problems happen and you can't see or hear me. So thanks for the introduction, Mark. As you said, I'm Riley. I'm a master's student here at OSU, and I am broadly studying bird window collisions. And I have several projects that I'm doing related to that. And today I'm going to talk to you about kind of my field project, which is testing window markers that have been designed to prevent bird window collisions, actually testing those in the field at some of our buildings here on our campus at OSU. So I guess I'll just start with a little bit of background on the problem of bird window collisions for those of you who may not know. This figure here is actually like 10 years old now, and it was showing that in the US alone, there's hundreds of millions of birds that die every year from hitting windows. Now, more recent research has actually shown, though, that the actual number in the U.S. is probably over 1 billion every year. And so when you extrapolate that to the rate that's happening across the whole world, it's billions and billions of birds dying every year hitting windows. Now, a lot of the research that has been done on bird window collisions, it really didn't start until the early 90s. Um, but I wouldn't really say that the topic of bird window collisions really became like a mainstream bird conservation issue until probably 10, 15 years ago. So a lot of the early research that was done on this was done in these controlled environments called flight tunnels, where basically a bird is released through one end into the tunnel, and then at the other end of the tunnel, there's two different windows or glass spaces, and one has a certain treatment on it, and the other one doesn't. And so it sort of gives the bird a choice of which window they're going to fly at. Um, and that was used for a lot of the initial research on different products used to or designed to deter bird window collisions. Now, in recent years, there has been a growing number of studies that have actually tested some of these products like in real world environments, not in a controlled, tunnel like this, but on actual buildings, on actual windows in the real world. And it's really, really important that we have these studies testing window collisions in the real world because they capture things that a study in a tunnel, even though those studies are useful. I mean, having a study in a real world environment captures things that you just wouldn't get otherwise. But there are certain problems or biases that are introduced whenever you are trying to study window collisions on actual buildings. And that's because typically the way that these studies are done is that you are literally walking around buildings searching for dead birds to sort of, that's your data is the number of bird carcasses that you find. And so there's a lot of kind of outside noise that gets introduced when you're doing that type of thing. For example, there's just inherent year to year variation. One year there may just be less window collisions than the next year. And that could be due to a variety of factors. There's so many outside factors that can influence window collision rates. And I don't have time to get into what those are, but feel free to ask me after the talk if you're interested. And then in addition to that year-to-year variation, there's also things like scavengers, here in Oklahoma, raccoons and skunks and cats who are constantly removing carcasses of birds that have hit windows. And then also, whenever you're relying on humans to walk around buildings and find carcasses, it's inevitable that you're not going to be able to find every single carcass. There's going to be some that you miss. And so some people might be, quote unquote, better at finding carcasses than others. These are all things that can kind of have an impact on when you're trying to estimate the number of fatalities, say at a university campus from birds hitting windows. These are all sources of bias that can sort of introduce some outside noise, I guess. And so the way that we try to sort of get around this is through what's called a before-after control impact study design, or BACI study design. And the general idea is just that if you have a treatment, so in our case, window markers, before the treatment, you have a period of monitoring where you have people going out looking for carcasses and calculating the number of collisions before that treatment occurred. And then after the treatment of window markers, you also have a field season in which you are looking at the collision rates at those windows that have the treatment applied to them, but you're also looking at windows that didn't have the treatment applied to them. And so what that kind of does for you is that it allows you to sort of have this overall idea of the trend. So if there was an overall increase in window collisions that year, or an overall decrease in window collisions that year. And so that can sort of help you to parse apart, like, if I have a decrease in window collisions at windows that were treated with these markers, is that actually because the markers prevented the collisions, or is it just that this year had less collisions than the previous year? So just a bit more specifically, the way that this worked with our study, we have these feather-friendly dotted window markers, which are just it's a grid of these white dots separated by about or an inch, sorry, separated by about an inch. Um, and the before treatment period, so before our treatment of window markers, there was a previous PhD student here at OSU who monitored collisions for several years, and so we're using his data as the quote unquote before period. And then after this treatment happened, which I should say that the treatment of window markers happened in the summer of 2023. So after that, we had our post-treatment monitoring season, um, in which we looked at the windows that were treated and compared those to windows that weren't treated. And then we did. I'm not going to really go into the details of this, but we also did trials to experimental trials to sort of get an idea of like for each surveyor, like how good were they at finding carcasses, and at each building, like in general, like how long did carcasses stay out before they were removed by scavengers or humans. And our study site, like I mentioned, is the OSU campus. The two buildings that we treated with the window markers were actually the two that were found to be the most dangerous, or the two that had the most collisions in that pre-treatment study. So that's the Noble Research Center and Gallagher-Iba Arena. Um, so at Gallagher-Iba Arena, we treated 4 glass facades. When I say facade, I'm talking about a continuous glass face like this. So it's not necessarily a window, but it's like a grouping of windows that is continuous. So at Gallagher-Iba Arena, we treated 4 facades, and at Noble Research Center, we treated 2 facades. And I'm not going to get into the data analysis stuff, but the important thing to know is that we basically, for each building, we calculated a rate of number of collisions divided by the number of surveys that were done at that building. And then we, using our data from those experimental trials where we, got an idea of which surveyors were quote unquote better or worse at finding carcasses, and the rates at which scavengers like cats and raccoons were removing carcasses. We also integrated that data into kind of our final estimate for number of fatalities that happened. So with all that said, I'll finally get into the interesting stuff, the results. Um, so the first thing that I want to draw your attention to is that if you look at this orange arrow here, the overall trend from the pre-treatment study to my post-treatment field season was that there was actually an increase in the overall number of bird window collisions at the OSU campus. But then, in contrast to that, if you're just looking at the windows that were treated with these window markers, those windows from the pre-treatment to post-treatment periods had a 67% decline in collision rates. And so what we're able to show here is that this, steep decline in collision rates was happening at the same time that there was an overall increase in collisions, which actually kind of means that, the actual decrease could even be greater than 67% because if you're accounting for the fact that you would expect that there would be an increase in collisions, I think that makes this decrease a lot more notable. And then some of you, if you were really paying attention, you may have noticed that I've been including a lot of pictures of Lincoln sparrows in these slides. And the reason for that is that Lincoln sparrow was by far the most frequent collision victim here. That's just kind of, a little regional thing. That's something that you wouldn't necessarily find in a lot of other places in the eastern or western US. You probably would find that in Wichita, being in a pretty similar environment here to Stillwater. Lincoln sparrows are, as I'm sure many of you know, one of our most abundant fall migrants. And so I think that is a big reason why they were number one. And then also some other species that we had a lot of sparrows, grasshopper sparrow, lark sparrow, and then also European starlings and mourning worms. So I think that the number one takeaway that I think we should get from this is that with our study design, we were able to sort of do what a lot of previous window collision studies haven't done, which is we showed that there was a decrease in collisions at these windows that were treated with the feather-friendly markers. And so we showed that they were effective in preventing collisions, but we also showed that at the other windows that weren't treated, that they actually had an increase in collisions. And so we're accounting for that overall trend. We're able to show basically to show that this decrease in collisions at the treated windows was even more significant because of that. And then we also were able to account for all the outside noise that gets introduced whenever you're doing window collision study, where we were able to account for the fact that uh, cats and skunks and raccoons and humans were removing carcasses, and we were able to actually incorporate that into our statistical analysis whenever we created these estimates. So going forward, this was just a study of one product, feather-friendly window markers. Um, there are tons of products that have been designed for window collision prevention. I'm sure many of you have probably seen some of them, or you know, decals and other window markers, and there's all sorts of products. And so we need studies like this for all these other products. And then I think that kind of what we've shown here is that we're never going to be able to treat every window in the US or every window on Earth. It's always going to be the type of thing where we are identifying, particular buildings or even particular windows or glass facades that are, dangerous or have really high collision rates. And then if we identify those buildings and treat those spots, that can actually go a long way in preventing, or sorry, that can go a long way in causing a decrease in bird window collisions overall, even if we're not treating every individual window or every individual building, because obviously that will never happen. It's just too expensive and too costly to do that. So with that, I would just like to thank OSU and the USDA for funding my research, and these individuals here for help with funding and fieldwork. And I will take any questions. Thank you.
Question. I've got a question. Can you hear me?
Yes, I can.
So you chose a window treatment using the dots on the windows at the exclusion of other treatments. What was your process for choosing that one?
So I actually didn't necessarily choose the product that we used. That was more something that was brought about by, this was a study that sort of the initial idea for it came about before I was here, and I was sort of hired to be the one who would collect the data and actually do the study. And so at the time that the funding was kind of identified for this project, I think there were different products that were considered. In the end, we chose feather friendly, I think, because a previous study here at OSU that had actually looked at window collisions at bus shelters. So we actually here on campus, we have bus shelters that are glass, covered in glass. And so there was a previous study that actually used those feather-friendly markers at bus shelters, and that was kind of the number one reason why we chose it, I think, kind of for continuity.
Okay.
Riley, did the buildings have more or less or the same amount of plantings in front of them or trees nearby? Is that something you could control for? Maybe you mentioned that.
It's not something that we controlled for, but you're absolutely right that that is something that has a huge impact. Um, and there were, I guess there were a few occasions in which that is something that could have affected the results. For example, between the pre-treatment period and the post-treatment period, there was a big construction project in which there was a parking lot that had been alongside one of the buildings. and then a huge construction project began where they were building a new building in that area. And so all of the trees that had been lining that parking lot were taken down. And, that is the type of thing that probably could have had an impact on, you know, maybe just less birds being around the building in the post-urban period. And, anytime you do window collision studies in a real world environment like this, there's just going to be so many outside influences that can, you could potentially explain part of the variation or part of the trends in window collisions. And I would just say it's impossible to account for all of them.
Were the birds that were collected the results of daytime collisions or nighttime collisions?
So we weren't necessarily able to tell exactly when the collisions occurred, but there has been previous research done here at OSU that has shown that the majority of the collisions here tend to happen like in the dawn to pre-dawn hours. That's not something that's necessarily the case everywhere. And so, the time of day or night that birds hit windows can vary depending on the region that you're in. But here in Oklahoma, it seems that a lot of the collisions tend to happen at dawn or just before dawn.
Riley, did the size of the window have any impact on your findings related to how many birds might have hit it? I saw the Coliseum, I saw the sports complex, huge panels of windows,
Yeah.
whole walls full of windows. But then I'm sure there's campus buildings or the bus stops that have a much smaller window surface. And in my house, we still have birds that run into the windows. Is there a size of window that's more attractive to birds?
I wouldn't say that there's a size of window that's more attractive to birds, but I would say that in general, the larger the window, the more dangerous it is to birds. In general, it's been shown by multiple studies in the past that at any individual building, the more of the exterior of that building that's taken up by glass, the more birds are going to be hitting it. So yes, that absolutely is something that we see with these huge glass spaces on the side of a sports arena or something, that they often are really, really, really dangerous for birds.
Thanks.
Are you able to estimate or just give a rough idea of how many birds would be hitting a window or some of these windows you're looking at per day or per week?
So it really, really depends on the window. On the windows, on an individual window, you say if we're talking about one of the ones that was really, really bad, I might find, at that window, I may find 3 or 4 per week if it's a really, really bad window. In general, for the majority of the windows, it's going to be probably 1 to 2 a week or 0. I mean, for the majority, it's 0, just because there's so many windows that I was surveying. But yeah, it's pretty variable, I would say. But I would say that the most you'd find per week at a really bad one is like 3 or 4.
Riley, I was wondering, they do sell decals and things, I know for sure, for window strike prevention, but curious about what you guys use in the future. If people are interested, is this going to be something that's sort of going to be on window manufacturers to have them include these types of things for the windows they put on a building, or still something that we personally, as individual consumers put on the window?
Well, I think there's a place for both. What I would say is that there are glass companies that are starting to kind of implement bird-friendly methods into new windows that they're creating. But that is very different from existing windows, and it's way more costly in a lot of cases to treat existing windows and not ones that are being manufactured. And so for existing windows, definitely, I think as consumers, we have a role to play there with treating, the windows at our homes even. I mean, if you think about it, you might think that the windows at your home, might not, you might see one or two, dead birds a year. And you might think, oh, I only see one or two a year. It's not that big of a deal. But if you consider how many homes there are in the US and then extrapolate that to the number of homes that there are, if all of the residential neighborhoods in the US, if people were treating their windows, that would have a huge benefit. So I would say that there's really a place for both glass companies, for manufacturing new windows and also for consumers treating existing windows.
Any other questions? Yep. All right. Thank you, Riley.
Thank you.
Great job, Riley.
Thanks.
All right, can everybody see my screen and hear me? I will take that as a yes.
Yes, I can hear you. Answer your screen.
Great. Yeah, hi everybody. My name is Holly Todaro, and I'm a PhD candidate at Oklahoma State University. And I'm really excited to be here today to share some preliminary results from one of my dissertation chapters that is examining the effects of weather and climate on avian reproduction. So we know that climate change, along with land use change, is one of the leading drivers of biodiversity loss across the globe. And this is evidenced by a ton of literature that's showing phenological mismatches, species ranges shifting, and populations declining. Now, across the globe, we also know that not all areas are experiencing climate change in the same way. We're seeing our higher latitudes experiencing more pronounced warming, while our mid-latitudes are experiencing more extreme weather events. Now here in Kansas and Oklahoma, we're seeing shifts in precipitation patterns, and we're seeing more extreme temperatures leading to droughts. And this has a lot of implications, negative implications, typically for agricultural production, as well as species that are in our natural systems. Now, many species are affected by climate change. However, birds face unique challenges as many aspects of their life history are intricately tied to ecological conditions. So for thinking about breeding birds here in Kansas and Oklahoma, there's only a small window of time when conditions are just right for reproduction. And as the climate continues to warm, this has serious implications for different metrics that are associated with avian reproduction, such as laying date, clutch size, and the survival of young. So when we're thinking about conservation and management in the face of climate change, it's really important that we're thinking about things like weather and climate and how that may influence these different metrics. Now, these relationships can become more complicated when we start thinking about other factors such as climate region, biome, and life history traits. So I'm not sure if you all are familiar with the game called Wingspan. It's really popular among the bird nerds and really popular within our graduate student community. But what you're seeing there on the left is a picture of the actual playing board, and it has different habitats or biomes that you can place your species into. And then if you can see the pictures of the bird cards on the right, you can see that these species have different life history traits. Some of them nest in cavities, while others may nest on the ground. Some species may be dietary specialists, while others are dietary generalists. So it's really important that we're considering these factors in the face of climate change because they may mitigate some of these impacts or may actually cause these species to be more vulnerable. Now, there's been a lot of really great research over the past 10 years or so that has tried to unravel some of these relationships. However, these studies have focused on a particular region, such as the North American grasslands. Maybe they looked at a certain weather type, such as precipitation, or focused on a single species, such as the great reed warbler. However, there hasn't been a study to date that has tried to synthesize all of this information on a global scale. And that is the overarching objective of this dissertation chapter. So I want to conduct a global meta-analysis that evaluates the effects of changing weather and climate on avian reproduction. And I also want to assess how those different metrics that are associated with avian reproduction may vary in response to weather across different climate regions, biomes, and life history traits. So I'm sure you're all sitting there, wow, Holly, that sounds like a lot of work. Trust me, it is. And so for the simplicity of today's talk, I just want to focus on a single analysis, which is investigating how an increase in spring temperature is influencing the timing of egg laying of all species across the globe. And I also want to know if species with different nesting types, if that may influence this relationship. So as I go through the rest of this talk, I'll be using these pictures that one of my undergrad researchers created to depict our ground, structured, and protected nesting species. So our structured nesting species will be those that nest in cups in a shrub, for example, while our protected nesting species would be species that nest in cavities. And so for folks that are unfamiliar with what a meta-analysis is, I want to walk you through what it is and some of the benefits of this sort of analysis. So a meta-analysis is really unique because it takes the results of individual studies and allows you to run an analysis on all of those results. So this is a really powerful way to identify trends and patterns in literature. And because you have a large sample size, it can lead to a lot of power and precision in your result. It's also a really cool way to explore the differences between studies, and it's an excellent way to provide guidance for future research and identify knowledge gaps in the literature. So it's a really powerful analysis. However, there can be some very tedious methods in doing this just to ensure that you're not being biased in any way and that you're getting the best sample of peer-reviewed literature that you can. And so the first step in doing that is coming up with a set of key terms that you're going to use to conduct a literature search. So after a lot of trial and error coming up with key terms, I was able to do a search in Scopus, which is just an online database, back in October of 2022. And that returned 3,251 peer-reviewed articles. And that was a ton of literature. So the first thing I did was I screened the titles and the abstracts, getting rid of anything that was not really related to my research question. And that left me with 659 articles that I had to read front to back. So because that is so many articles to read, for today, we're just going to focus on a subset of that data because I'm still reading papers. So we're going to be focusing on 23 papers from that data set. But because some of the papers include analyses on multiple species, I actually have a sample size of 37. So that's not terrible for some preliminary results. So from these 23 papers, I extracted information related to spring temperature and the timing of egg laying. I also extracted information related to site, such as location. I also extracted information related to species, what species was it, and what sort of nest did they construct. And then, of course, I extracted any results that were related to this relationship. So with those results, I transformed them into a Pearson's correlation coefficient just to make things more comparable across studies. And it really allows folks to visualize the results in an easier way, which you'll see in the coming slides. For any of the stats people out there, I'm not sure if anybody's stats savvy in the audience, but I ran a random effects model in the metaphor package and program R, and I used species as a random effect to account for any variability between species in this data set. And then, of course, I included nest type as a moderator or a predictor variable to try to understand what variability was causing the differences we were seeing in the results, with the expectation that our ground nesting species would be more negatively affected by that increase in spring temperature change because they may be more exposed to these temperature differences. And this map on the right here is just showing the locations of the 23 studies that are in this small data set. So interestingly, just the random 23 studies that are used in this analysis, they're mostly in Europe. You can see there's a couple studies in North America. The larger the circle, the more data points from that particular country. I know right now that the data that I've collected so far, we have a lot more studies in North America as well as South America, but we're definitely missing studies in places like Africa and Asia. So these are the results from my meta-analysis. And so what we're looking at is a graph that's going from -1 to 1. And that red dotted line at 0 indicates that there is no effect of temperature on egg laying. If we see a negative value, that means there's a negative correlation between spring temperature increases and egg laying. And then anything on the right of that red dotted line would be a positive effect of temperature on egg laying. So up top, you can see all species, regardless of nest type, we're seeing a negative relationship. So what that means is that as the spring temperature is increasing, all species, regardless of nest type, are laying their eggs earlier in the spring season. Now, I wanted to break that down with the different nest types. So you can see that our ground and structured nesting species are more negatively affected by this increase in spring temperature than our protected nesting species at the bottom there. And you can see that within our protected nesting species, we're seeing a lot of variation in responses. Some species are responding negatively to this increase in spring temperature, while others may be not really affected or maybe positively affected by this. And I wasn't surprised that there was going to be some differences between our protected and ground nesting species, for example, but I was surprised by the variation in our protected nesting species. So one thought I had was potentially there was a difference in like the thermal properties between a human-made cavity, like a nest box, compared to a natural cavity, such as a tree. So I ran some statistics, specifically a walled type chi-square test, to see if the differences in cavity type was causing this variability. But unfortunately, with this data set, it is not. So I'll have to investigate this a little bit further, and hopefully with more data as I read more papers, this result will maybe become a little bit more clear. So to wrap things up, with the increase in spring temperature, we saw earlier egg laying for all species regardless of nest type. And we saw more significant negative correlation for our ground and structured nesting species. As I mentioned, this is likely due to the direct exposure to environmental conditions that our ground nesting species are experiencing, and that can lead to things like accelerated egg development. Similarly, our structured nesting species are likely more subject to ambient temperature changes, whereas our protected nesting species may be less affected because they are protected from that increase in spring temperature. So what does all of this mean for our local and regional conservation and management efforts? You know, here in Oklahoma and Kansas, there are a ton of ground nesting species, especially our grassland species. And I think one thing that we can do as conservationists and managers or, you know, just within our own properties is providing thermal refuge for these species, particularly with native vegetation, with a diverse vegetation structure to provide them some protection from this extreme weather. And as I mentioned, this is an ongoing project. So in the future, I'm hoping to increase the number of studies within this analysis and others by reading more papers. I also want to account for phylogenetic relatedness among species. If we think about a phylogenetic tree, species that evolved more close in time, they may react more similarly to this increase in temperature, for example, as opposed to species that maybe evolved earlier or later. And then, of course, I'll be conducting additional meta-analyses looking at different combinations of weather types and reproductive parameters to understand really how weather and climate are influencing avian reproduction. So, for example, I'll be interested to see how things like precipitation, for example, is influencing nestling survival, how sea surface temperatures influencing productivity, just to name a couple, but I have a huge list of analyses that I'm hoping to conduct. So keep an eye out in the next year or two for some pretty cool research. And with that, I want to thank y'all for showing up tonight in the snowy weather and listening to me. I also want to thank the Fritz L. Knopf Doctoral Fellowship Program in Avian Conservation, as well as the South Central Climate Adaptation Science Center and the Natural Resource Ecology and Management Department at Oklahoma State University for research funding. And I also want to thank Rachel Folk for her assistance with data collection. And with that, I'd be happy to take any questions.
When you did your studies on temperature, did you find that laying earlier in the season due to increased temperature had either a beneficial or a detrimental effect on the birds that had hatched as it relate to blossoming of flowers or larvae that are beginning to hatch, available food sources, anything like that?
Yeah, I think it was a mixed bag of results, sort of depending on what the paper actually studied. You know, some of the papers just looked at temperature and egg laying and didn't actually like follow the nests. Like they didn't report whether that nest, when it hatched or if it survived. But I would say in most cases, the papers that I read, the species that did lay their eggs earlier, they did fledge. Young that, so they weren't affected by this increase in temperature in terms of survival. But it depended on like how high the temperature actually was. If we were seeing extreme temperatures, some of those eggs were no longer viable. So it just really depended on things like, habitat type, climate region. And those are things that I'm hoping to explore a little bit further when I have more data.
Thank you.
Yeah, thanks for the
Holly,
question.
Holly, with this meta-analysis, is there a model where you could utilize AI to cover through so many articles that would assist you in your studies?
That is a great question. I have used some AI to go through the like initial like 3,200 papers or whatever it was to like eliminate duplicates. And like I was getting a lot of papers that were related to like meat chickens, which obviously aren't really important to this research. So I was able to pull those out so I didn't have to. But in terms of actually like pulling out the results, to my knowledge, there's nothing that is like that accurate that can pull out all the results that I need. It's kind of tedious, all of the information that I need to extract. So for the most part, I read all of the papers, or I had some really awesome undergrads that helped me collect some of the data.
Well, it would seem to me that you could scan all those articles
Yeah.
and then have AI do all that tedious work for you.
Yeah, that would be great. Hopefully they can come up with something that is, 100% accurate in the future.
Yeah, thank you.
Holly, I was just wondering if, along with increasing warmth earlier in the spring, is it also possible that there's increasing variability in the spring temperatures, so that maybe like if you nest earlier, that puts you at danger because then it could all of a sudden get really cold again?
Yeah, of course. Yeah. A lot of those papers are focusing on things like phenological mismatches. And so those papers weren't really included in the database that I was reading. But yeah, I mean, with all this variability and cold snaps that we're seeing, could definitely be detrimental if our species are arriving earlier, laying their eggs earlier, and then we're seeing these cold snaps or really cold temperatures could definitely be detrimental.
You mentioned that you were going to look at the phylogenetic analysis part of it. As far as the species that you had in your current section you've done research on, are they mostly songbirds or more, are there like shorebirds mixed in there?
Yeah, so I think within this data set, I had a pretty good mix of like passerines, seabirds, and shorebirds. I have in my overall database, I think I have around 400 species, which is awesome because if, I only had like 100 species, a lot of the results may be due to that phylogenetic relatedness. But what I'm seeing so far is that I have such a large database with so many species that that's not influencing the results.
Seems like there should be really interesting things to come out of that.
Yeah, keep an eye out.
Do you think the birds are responding to temperature? Or do you think they're responding to the changes in the plant community that is responding to the temperature? Or is it all so tied together that you can never separate it?
Yeah, I mean, I think the birds are like in terms of laying date, I think as we're seeing an increase in temperature, that's triggering them to, lay their eggs earlier in the spring season. But, as the birds are migrating, we are seeing things like phenological mismatches and, when they're arriving to the breeding grounds, for example, and the plant, green up may have already occurred or they're missing the emergence of insects. So I think kind of to your point, a lot of it is really interconnected and it can be kind of hard to parse out like what exactly is causing some of the effects that we're seeing. So yeah, it's definitely complicated and I'm hoping that some of these results can make that a little bit more clear.
Any other questions?
Thank you.
Thank you, Holly.
I have a question. Can you hear me there?
Sure can.
So it seems like it's already very complicated, but you could get it a lot more complicated by having tropical versus migratory, neotropical migrants versus our resident birds, like an even house wren versus a Carolina wren, or on the ground nesting, whether it's in a marsh or at grasslands. There's all kind of ways you can break that down even more complicated.
Exactly. Yeah. And that's sort of the plan. Like I said, I think in my overall final data set, I have about 400 papers and I'm running some preliminary results or preliminary analyses on some of the other combinations. And things like habitat type and climate region are showing to be important in some of these relationships. So hopefully somebody can take my results, which is pretty broad, and maybe apply that to more regional or local area to maybe parse apart these relationships a little bit further. So,
All right, thank you.
yeah, thanks for the question.
All right. Thank you, Holly.
Yeah. All right, Dylan, I think I stopped sharing my screen, but I'm not sure.
All right, well, let's see if I can get mine. Okay, can everybody hear me and see the presentation?
Yep.
Yes.
All right. Well, my name is Dylan Cooper. I am a PhD student at Oklahoma State University, and today I'm going to be discussing a citizen science project focused on shrikes. The first chapter of my dissertation, I am looking at habitat use by loggerhead shrikes across the state of Oklahoma, and I'm analyzing that in an occupancy modeling framework. And that study is going to be unique because I'm combining so many different data sources that have been relatively untapped. I'm looking at atlas data, breeding bird survey data, Christmas bird counts, and then I'm also doing my own surveys. And when I'm doing my own surveys, I'm finding all of these prey items that are impaled by loggerhead shrikes on either barbed wire or a thorny plant. So that's really sparked my interest in this bird's diet. And there is a citizen science project that engages the community in these types of questions. So that project is Larder Locker, and it's housed within the iNaturalist database. So if you have an iNaturalist account, you can join this project. And if you happen to find, as you're driving along, you see these prey items that had been placed on the barbed wire by loggerhead shrikes, you can take a picture of it as an iNaturalist user and upload your picture to this project. That type of information will help scientists better understand where the shrikes were, so what habitat associations they're using, in addition to the diet, what they're eating. To my knowledge, there hasn't been a study yet that has looked at shrike diets across a very large scale. Like I'm thinking of like USGS regions, so like the entire Southeast, the entire Southwest, which Larder Locker would present a unique opportunity to be able to do that. So that's what's really exciting to me. There have been a lot of studies on shrike diets, but they tend to focus mainly like in one state, for example. So if you don't know what a loggerhead shrike is, it is a smaller songbird that has a diet similar to a larger bird, like a falcon, for example. And because they lack those strong talons that a falcon has, they have this unique behavior where they actually impale their prey. And that is the area where they impale their prey is called a larder, and each prey item placed at the larder is called a cache. So what you see on this picture here is a loggerhead shrike caching a prey item at its larder. This type of information that can be shared on Project Larder Locker is important because the loggerhead shrike is declining across its range, and it's not entirely understood exactly like why they're declining everywhere. Habitat loss is a big thing that's implicated in the decline, but there are other factors as well, including pesticide use and how that could potentially affect the prey that's available to the shrikes in that part where the pesticides are being used. So for my presentation today, I'm going to go over kind of like what data is available on Larder Locker and some of the emerging trends and biases in this data set. I'm going to separate these into trends with user participation, trends in how the data is spread across the US, how the data is parsed across different seasons, different months, and then also how the data is spread across taxonomic groups of prey. On the right side of the slide, you can see a picture of what the project page on iNaturalist looks like. So Larder Locker user participation. There are now 200 users that participate in this project, with the majority of them being casual users. So of those 200 users, 131 of them have only uploaded a single observation to the project each. This participation varies widely. So I made this histogram, and you can see there's one user who contributes over 16% of all the observations currently added to Project Larder Locker. And there are only a few users who've contributed more than just a single observation. And then if we look at the spatial distribution of Larder Locker observations, this kind of ties into the bias in user observations. For example, the one user who has over 16% of observations on Project Larder Locker comes from Alabama. So that kind of explains why that particular state has more data compared to other states. In Oklahoma, we have so much data because we're out in the field looking for shrikes, and we're finding their larders and posting it to Project Larder Locker. And then in Arkansas, there's a PhD program that they study shrikes there as well. And the one of the PhD students at Arkansas was actually the one who created this project on Larder Locker. So some of these places that have a lot of observations is because people know about this project and they're studying shrikes in that region, for example. So we have over 80% of observations come from the southeastern US, USGS region, and over 70% of those observations are from only four states: Alabama, Arkansas, Oklahoma, and Texas. One reason why I'm really excited to be giving this presentation today is because on Larder Locker, there's only one single observation from Kansas. So it would be really great to kind of increase participation and interest in this project that people may not know about in Kansas to start generating some more data from other states that that type of data isn't really represented in the database as it currently stands. temporal distribution of Larder Locker observations. So the majority of the observations, 62% of them, are from the non-breeding season. And I just kind of was being very generous about that term. So I just separated the year into 6 months of breeding season, 6 months of non-breeding season, which might not exactly reflect when the shrikes are either breeding or non-breeding, but just to kind of give some idea of where that data is spread between. So, um, and then many of the observations, so about 34% of all observations come from November. And that is because recently, when we were doing our first round of winter surveys in Oklahoma, we just found a ton of shrike caches in November. So that really skewed the data from the last time I gave this presentation and looked at it. But even before, more of the observations were from the non-breeding season than the breeding season. So we do need a more balanced sample and we need more data from the breeding season. Now I'm going to look at kind of the taxonomic distribution of larder locker observations. So what types of prey items are people finding at the shrike larders and posting to Project Larder Locker? And I've kind of separated this by region. So the majority of our data is coming from the southeast. So we have a pretty robust sample size here. As far as the breeding season goes, this is pretty consistent with historical data that has looked at shrike diets where a majority of the diet, over 50%, consists of arthropods and especially beetles and grasshoppers. And then when we look at the non-breeding season, again, I have to stress that this data is biased towards the extreme amount of November observations that we had. So that might explain why the proportion of grasshoppers was so high in the non-breeding season, because this might have been when it was still warm out and we were finding all of those grasshoppers in November. Now it's really cold, so there probably wouldn't be as many. If I were able to kind of separate the data out into smaller bins where maybe winter would just be December, January, February, I think we would see a higher proportion of vertebrates and especially rodents in the diet. But unfortunately, even with the Southeast having the largest samples out of any of the USGS regions, there's still not enough data available on Project Larder Locker yet to separate the data into these smaller bins and have it be meaningful. So as Project Larder Locker grows, that will be something that will become feasible and provide a more accurate picture of how shrike diets are, or at least the prey items cached at larders are changing across these seasons. Next, we have the Southwest. So we do see that there's kind of this similar pattern where grasshoppers and beetles are making up a majority of the summer diet. In both regions, actually, beetles did not comprise a lot of the diet in the non-breeding season, and that's probably due to the phenology of beetles. They're probably not as abundant in the non-breeding season compared to the breeding season. So that was kind of a pattern that was preserved across USGS regions. As far as the non-breeding season, this data from the Southwest is kind of more like what I would expect to see, with the diet kind of shifting from a majority of arthropods to vertebrates becoming more important in the shrike larders during the non-breeding season. So we see that increase in rodents found at larders in the non-breeding season. Compared to the previous slide with the Southeast, frogs made up a larger proportion of pictures posted to Project Larder Locker. In the Southeast, there were not as many pictures of frogs being posted from the southwestern states, but there were more reptiles, and that probably reflects maybe like the aridity of the environments in the Southwest and the Rocky Mountains states, like we're going to look at next. The frogs are probably more ephemeral when there are, after like monsoon rains, when there's more of that type of environment available. But in the southeast, those are states like Florida, where there might be more types of habitats to sustain those populations over a longer time. So they might just be more abundant in that type of habitat. Next, I have the Rocky Mountain states. So again, we see that pattern during the breeding season where the majority of the diet is arthropods and especially grasshoppers and beetles. And then in the non-breeding season, there was a very small amount of data, so there was only 9 data points for this. So we're really lacking for the non-breeding season data in this region, and that's probably because the USGS defines the Rocky Mountain states as Colorado, Wyoming, Utah, and New Mexico. And in 2 of those 4 states, shrikes really are like a summer breeding resident, and they probably migrate to further south in the winter. Shrikes are resident in New Mexico and Utah throughout the year, though, so that's where those non-breeding data points would be coming from within the Rocky Mountain region. I did not look at the Northeast region or the mid-continent region, which the USGS classifies as Kansas. And that would be just because there wasn't enough data from those regions. In the Northeast, we do have some data from Virginia where loggerhead shrikes are present. And in the mid-continent, we did have a few data points. We had the one observation from Kansas, but there just wasn't enough. So we really need better representation from those regions and more engagement and participation in Larder Locker from people living in those regions. I wanted to give an example of what could be done with Larder Locker data. So I have begun to map loggerhead shrike larders in Oklahoma. How I did that was I extracted the data from Larder Locker, which I was able to export all of the observations, and I was able to, each observation has a coordinate pair associated with them. So I was able to map out those coordinates, and I combined any coordinates that were within a 10-mile radius of each other. They were combined to a single point on the map, and the numbers on the map represent the number of caches that were found at that particular larder. So this map can be really helpful because it will help me to be able to continue monitoring those larder locations that I have been finding in Oklahoma. And kind of the next steps, what I'd like to do is I'd like to sample insects around the larder location that are available in the environment and compare those proportions to the prey items that we're finding cached at the larder. Because a few studies have actually looked at prey selection by loggerhead shrikes because these like the studies that have been done, they document what the loggerhead shrikes are eating or caching at their larders, but they don't actually sample what's available in the environment to compare it to. So we don't really know if loggerhead shrikes are selecting for a particular species of grasshopper or a particular species of beetle. So I want to see if the proportions of any particular species of those arthropods are more abundant at the larder than they are within the environment, and do some analyses to see if that might maybe if they are selecting for a particular prey species of grasshopper or beetle, is it because those confer more protein content or lipid content and perform some of those analyses. So that's where I'm hoping to go to next with the larder data that I've begun collecting and documenting on the Larder Locker project. I wanted to end with some eye-catching catches that can be found on Project Larder Locker. So at the top left of this slide, we see a strip of bacon that was in Louisiana, and that observation was actually published in the journal Southeastern Naturalist. So you can read that paper if you're interested in that one. And then the Popeye's chicken that was observed in Alabama, and this was at a location where and there was an active shrike nest. So our observer from Alabama, who has all those observations, she was able to kind of like document this nest throughout the season and watch them catch the prey items from the Popeye's chicken. And in some of these regions, like especially far south in the southeastern regions, like we don't see them being shrikes as being very urban in Oklahoma at all. But in Texas and further south, they can be pretty very urban. So I thought those observations were cool. I included a picture of a Costas hummingbird that was impaled on a cactus because I just thought it was really cool that a shrike would be able to catch something as fast as a hummingbird. On the other end of the spectrum, shrikes have been able to capture and impale birds that are even larger than them. There was a recent article in the Wilson Journal about them impaling whales. I think the largest bird on Larder Locker was a mourning dove. I included an observation of an engorged tick that was observed in California because I thought that was a really cool one. A picture of a blister beetle at the bottom left. Those blister beetles have these like chemical compounds, so not a lot of predators are able to eat them, but I thought it's cool that the loggerhead shrike can. I believe there's a review paper about loggerhead shrike predation on blister beetles. And then I also included a picture on the bottom right of a fiddler crab because I thought it was cool that the loggerhead shrikes were eating seafood in Texas. That is my presentation. I've included a QR code here. So my hope is that maybe some of you might be interested in joining Project Larder Locker and or maybe like telling the people that you know in your communities about this project and seeing if some of those people might want to join. So the next time that you're driving along and you might be in an area where there's a lot of barbed wire fence, like I would just encourage you to maybe like slow down, see if you can find any cool eye-catching catches. And the next time I present this, I've been trying to like promote this project a lot. Maybe your picture will be featured on the eye-catching caches. Thank you for your time. Let me know if you have any questions.
I have an observation. I find it amazing that you can say Larder Locker that many times without totally getting tongue-tied. So,
It is a bit of a tongue twister, isn't it?
and then I have a question. You mentioned diet and the words Larder Locker refers to food, and it's assumed that they're catching these things and caching them for a food supply. And yet in November, a bunch of things are still impaled as opposed to having been eaten. To what extent have studies been made to show at what rate these foods are eaten? And that brings up another area that I had heard before, that perhaps the whole purpose of impaling is not so much for a food supply as it is to impress a potential mate, as with hunting skills and the ability to care for chicks in the nest, and may not be as important for food as it is for mate selection.
That's a good observation. I definitely did read one paper that mentioned shrikes were like the rate of return to their cached items wasn't extremely high, and that and there have been studies that show that like the amount of prey items cached at a larder does increase the shrike's fitness. So it is definitely playing a role in mate selection, like to attract a mate.
And then this is just my lack of understanding of loggerhead shrikes and what they do. Do we know how often they do impale things? Is that the main way they have to kill certain things, or can they just dispatch prey and just eat it right there?
Yeah, so there was a study in Arkansas actually that and I believe that one was in Southeastern Naturalist as well, that compared the prey that shrikes were catching and eating immediately just by watching the shrikes, and then comparing that to the prey that was being cached at the larder. So what that study had found was that like they are eating a bunch of smaller prey items that are, you know, they just consume it immediately upon capture that will never show up in a larder, for example.
Interesting.
I can only think of once that I've ever seen a shrike cache, although I haven't gone looking for them, but I find it amazing enough to find a shrike.
Yeah, it can be challenging to find them too because they are declining. I have found that as I've been like surveying for shrikes and like starting to document things on Project Larder Locker, I'm finding more shrikes now than I was before. So I think like also just kind of like developing that search image can also kind of help in like maybe if you start to like get into like documenting things that you do find, if you do end up finding anything, then I think that it can be kind of surprising how like once you find one, it kind of snowballs and you end up finding like starting to find more.
Yeah.
My wife will probably slap me silly for saying so, but I recently read where a guy divides seasons into six: winter, mud, spring, summer, fall, and stick. And I wonder if it isn't possible that during stick season, the larders are simply easier to find.
Yeah.
Yeah, I think that could be something too if the larder is like located on a plant instead of like barbed wire. I think those ones would definitely be easier to find when the plants don't have leaves. So I think there is something to that. In my region, I've only ended up finding a few larders like on plants. The vast majority of the larders that I end up finding in Oklahoma are on barbed wire, but that also could be related to the detectability.
In terms of using it for storing food, would you expect that they would do it more when times when food is short? There's not as much food around.
Well, that is a potential hypothesis. The paper that I had read that was talking about how they didn't have like high return rates, and that, but they ended up finding that the larders that were more populated with prey items, they had like higher mating successes. That paper also talked about how they did observe actually the juveniles feeding on the larder items. So the other thing that they had mentioned was that they thought that like during this period of time where the juveniles are fledging and still learning how to hunt, and maybe they can't capture as much prey on their own, that the prey items at the larder might be important for that particular life stage. So I thought that was really interesting, cool, a really interesting observation as well. So I think that that could be something too, is that like they're maybe during the breeding season, they're kind of like stockpiling these larders to attract a mate, and then also their young might they might kind of save it for their young during that critical time. But I think that it's possible that they could be using them like during times when it's like really cold and they're not finding much prey and coming back to them. It just doesn't seem like the return rate was very high in the one study that I had read.
We'll put your QR code in our newsletter, and maybe we can get more people looking.
Awesome. I appreciate that very much. It would be great to, yeah. I really enjoy like doing this project and hopefully being able to recruit more people. Think that that would just be great. And that was really my goal presenting today, was to promote this project and increase the participation in it.
Yeah, it's a species not that many people know about unless they're bird watchers, and it's declining.
Yeah.
Any other questions for Dylan? No, thank you, Dylan.
All right. Thank you.
Yes, they probably won't join us for snack.
We have cookies. Thank you guys so much for doing this. I
Yeah.
appreciate it.
Thanks for letting us present and thanks for showing up.
Yeah.
Too bad the weather didn't cooperate, but I guess that's the way it goes.
Hope everyone has a safe drive home.
All right.
Yeah, thanks to everyone who actually made the drive out to the nature center. That's awesome.
That may say something about our mental acuity more than.
Thank you.
It made it a memorable evening, I think.
Thank you.
Thank you.
We'll stay in touch. Good to meet you all.
Good to meet you too.
Could have gotten worse. Right?
It could have
He
been
wasn't
much
supposed
worse.
to do anything.
Thanks everybody else for being online this evening.
Yeah.
We're gonna go have cookies.
And we will see you next month.
Thanks all.
Which is another virtual presentation.
Oh, we gotta figure out how to do that. I think it would be interesting.
For all those in the room, thanks for putting this on, saving us a trip out into the weather.
Put a ring doorbell.
Oh yeah, you're welcome. No problem.
Stop recording.
Why not fight it rather than put them right out there in the open where